today is first is the Lpcb. So Lpcb of dermatophytes. Okay. So you all have just had a class just now. Sorry. Just had a class on the superficial the fungal infections and the subcutaneous fungal infections, and you've learned about the theory, the different types of fungi that cause the diseases, and which layer of the skin and the subcutaneous tissue which particular fungi is affecting. So now, just for demo purpose, let us look into what is Lpcb. So Lpcb is lactophenol cotton glue. So what is lactophenol cotton glue? It is a type of a stain which stains the fungi, and it helps us visualize it on wet mount. ठीक है? So जिसमें जो lactophenol portion basically acts as a disinfectant. It acts as something that will keep the culture moist, because if it becomes dry, then there's no point of making a wet mount and we will not be able to see the morphology of the fungus. And cotton glue is basically a dye that gives this characteristic blue color to the fungus or the fungal structure that we have to see. Okay. Right. So let us all review. First thing I want to understand, if you all know this, what is the structure of any fungus, any dermatophyte? Because there's no point of me discussing the staining if you all don't know what is the structure of the dermatophyte. So this is a hyphae. Dermatophytes have got septate hyphae, and as you all have learned, there is Trichophyton, there is Epidermophyton, and there is microspore. Okay. So Trichophyton usually does not have any macroconidia. It has characteristic septate hyphae which are very thin, and it has microconidia that are arranged in certain arrangements on the hyphae, by which you can identify them on Lpcb. So to make an Lpcb mount, let us just briefly discuss this. So Lpcb mount बनाने के लिए what we do is we take a glass slide and we take a drop of normal saline in which we add the culture isolate, and then we we take a drop of the Lpcb and we add the culture isolate, and then we tease the culture. What do you mean by teasing? Teasing means we take two teasing needles and they separate the structures. Now coming to that structure, these hyphae, they are usually in the form of mycelium. I hope you all are aware of the term mycelium. Mycelium means a collection of hyphae. So everything is jumbled up. It will all be like this in culture. Everything will go together. and you will not be able to understand what is the structure. When you tease using two teasing needles, these hyphae get separated. Okay, so when they get separated and you have LPCB in the wet mount, then you are able to see them clearly in the microscope, 40x of the microscope. See, 40x magnification. Okay, so after you have teased the culture, then you place a cover slip and then you observe it under 40x. So what do you see now? So basically, if we are seeing Trichophyton, Trichophyton causes infection in all three: nails, skin, and hair. Okay. So in case of few, I will mention the very common species that you all at undergraduate level should know. So we have one that is Trichophyton mentagrophyte. Next we have Trichophyton rubrum. Okay. Then we have Trichophyton Scholleni. Okay. Let us concentrate on these three very common species. If you don't remember the species also, it's okay. But they have characteristic features and they come as questions in your exam. So what's so special about mentagrophytes? So Trichophyton mentagrophyte has got very thin septate hyphae, and these septate hyphae have got clusters of microconidia along it, and this starts looking like grapes. Okay, these microconidia. So this is a rare structure which with structures of microconidia as in form of grape clusters, and this is seen in Trichophyton mentagrophyte. Okay, macroconidia is very rare. Okay, now in case of rubrum, again there is septate hyphae. ये जो blue color में I'm drawing specifically so that you will understand that in the LPCB mount you will be able to see the structures just like this. So we see the microconidia arranged linearly along the hyphae, and this appearance is known as birds on a wire appearance. Now, what is another special thing about rubrum is that rubrum is a red color. Basically, it is this culture on the reverse side of the tube will form a red color pigmentation. Okay, every SDA tube has got a obverse side and has got a reverse side. When you observe the culture, this you have learned in lab diagnosis. So in case of rubrum, it has the red color pigment characteristically. So when you are given these tubes in Grand Viva tray, you will be able to see the colony morphology, and on the reverse side, you will be able to see the pigment, and you have to report it in the same way in the exam. And on the now, since the reverse is red, in the obverse side, we will be able to see a white cottony colony, velvety cottony colony. Okay. So now moving on to another Trichophyton, which is Trichophyton scholleni. Trichophyton scholleni, as the name suggests, it has got something known as a favic chandelier structure. Okay, so favic chandelier matlab the hyphae is like this, somewhat like this. Okay. So this looks like a chandelier, so that is why it is known as fabric chandelier hyphae. This is for the hyphae, not for macro या macroconia. Right? So now the second is Epidermophyton. So Epidermatophyton, as the name suggests, affects the epidermis, that means the skin, and it affects the hair. Okay? So in Epidermatophyton, you don't see any microconidia is very rare, but there are very characteristic macroconidia that you will be able to see, and these are known as club-shaped, thick-walled macroconidia. They look like this. They are double-walled. They have septa in between. Okay? They arise from a common point on the hyphae. So you will be able to see big, large macroconidia. They will arise from a thin septate hyphae. And this is Epidermatophyton. Don't need to remember the species, but you should definitely remember that it looks like this. It has got a club-shaped macroconidia. Okay? The colony is khaki color. ठीक है, तो वही colony उठा के you mix it with lactophenol cotton glue and then you prepare a wet mount, you will be able to see these structures. Then moving on to the third dermatophyte, that is microsporum. Microsporum, as the name suggests, is micro to फिर hair and nails. Okay, so microsporum, basically you need to know only one or two maxima species. Microsporum canis. This is also a zoonotic infection acquired from cats, mainly seen in places like Brazil, because there is a fashion of the owners usually kiss their cats. So that is how they acquire the infection via respiratory route. Okay, the second is M, that is microsporum hoki. Okay, so in canis, these are all identified with the help of macroconidia. Okay, so इसकी macroconidia is spiny. By spiny, I mean you'll be able to see an irregular surface like this when you are seeing it on Lpcb. It is thick-walled and it has these compartments, transverse septa in the macroconidia. Whereas cookie and cookie has got a double-walled, very clear double-walled macroconidia with septa in between. Right? Okay, so this is basically how on Lpcb the fungus will look, the dermatophytes will look like. But if there is any question, you all can please ask in the question box. I see Prakhar has replied. Red pigment. I think you have spoken for the this Trichophyton rubrum. Correct. It gives a red color on the reverse. You all can please post your questions in that question box, and I will simultaneously answer it. And कुछ अगर clear नहीं है, if you feel that it's not clear, then I can re-explain it. Then we quickly move on to gram staining of gram-negative bacteria. Okay. So if there are no questions, then what I'll do is let us move on to the gram staining of gram negative bacteria. I think तुम लोगों के लिए class I think मैंने ली थी gram staining वाली one session, I think batch A and batch B. So let us quickly review the steps of gram staining and the importance of each of the step and why are we doing it. So the first step, if anyone can write in that question box, is what in gram staining. And I clearly remember telling you all कि क्या बताना है first step में और क्या नहीं बताना. Very good. Prakhar, very good. You have to heat fix smear preparation. Yes, yes. बस प्रखर ही है पूरे क्लास में. Others are not getting any attendance. So yes, as correctly mentioned, first we take a slide. This is a glass slide. We put a drop of normal saline or distilled water. Can anyone tell me if we are supposed to use distilled water or normal saline? बताओ जल्दी बताओ question वाले box में please write your answers. And this is an important question you all ask. Kartike, why do you think it is normal saline and not distilled water? Okay, either Prakhar is saying either. Okay. Okay, so we are using normal saline. Distilled water में ना it acts as a desiccant. The bacteria might get lysed. ठीक है, तो हम normal saline use करते हैं. We take a drop of normal saline. Wait, just yeah. So we have taken a glass slide. We take a drop of normal saline on this. We take the colony now from the culture plate. अब मेरे पास ऐसे बहुत सारी colonies हैं, ठीक है? So many colonies I have separated. We pick one part of the colony with a very thin straight wire. We pick only a part of the colony and we emulsify it in normal saline that we've taken on the slide. ठीक है, तो बन जाता है एक smear. This smear is allowed to air dry. So once the smear air dries, what we can do is we will heat fix it. What are the other methods by which you can fix a smear? If I do not have a burner, what do I use? You can alcohol fix a smear also. Alcohol on, ठीक है, alcohol से भी आप fix कर सकते हो, formalin से भी fix कर सकते हो. So then that is known as chemical fixation, right? So you have your slide, you have your smear on it. ठीक है, the first step is crystal violet. So first thing is crystal violet. Crystal violet is also known as the primary stain. This method is the modified Hucker's method. ऐसे तो gram stain was invented. The method was invented by Hans Christian Gram in around 1980s. Okay? And then there was a lot of modification. This is what we are using in our lab also. Crystal violet डाला and you keep it for 2 minutes. This is the primary stain. Where will it bind? Remember the structure of the cell wall of the gram negative and the gram positive organism. So as you remember, in gram positive organisms, the peptidoglycan layer is very thick. There the crystal violet goes and binds to that peptidoglycan layer. And in gram negative organisms also there is a peptidoglycan layer, but that layer is very thin. So we will come to know what happens in that layer. So दोनों में जाके crystal violet has gone and settled. Next we are adding a mordant, which is iodine. It is also known as grams iodine, consisting of potassium iodide and iodine. Okay, so this iodine goes into the peptidoglycan layer and then forms a complex. This is an insoluble complex, water insoluble complex. And then what happens is, when we wash the slide in the next step, grams iodine is to be left for 2 minutes. ठीक है, उसके बाद what we do is, when we wash the slide with acetone, we don't wash it basically, we just run the acetone over the slide such that it touches the slide or touches the smear for that matter, 2 to 3 seconds only. So this complex that is deposited in the thick peptidoglycan layer will not get washed out in gram-positive organisms, whereas it will get washed out in gram-negative organisms. Okay, because the layer is thin. Then, as a counter stain, we are using 0.25% saffronin. ठीक है, तो what it does is कि जो gram negative organisms की peptidoglycan layer में से the complex has been washed out, saffronin will go and stain those gram negative organisms and you will be able to see them as a pink color as I've drawn here against the background. ठीक है, so any gram negative bacteria on gram stain will appear as a gram negative rod. ठीक है, this is like a very highly magnified image. Okay, so in the exams, what do you do? How do you report a gram stain? In the exams, you will write, supposing you see this, okay, in the microscope after staining. So you will write gram negative bacilli seen with parallel sides and round ends, as you can see here. Okay, because तुम लोगों को exam में you will mostly get members of the Enterobacteriaceae family, ranging from E. coli to Klebsiella, Pseudomonas, Salmonella, या Shigella. So they have this very constant structure on gram stains, so you can use this statement everywhere. Okay, rounded ends and no particular arrangement. The bacilli are uniformly stained. Each word in this sentence has got a huge importance. Gram negative bacilli, this is the gram reaction. Bacilli is clearing the fact that you have rods in your smear. Parallel sides are important because there are curved bacilli that you all learn about. If you all remember, Vibrio cholerae is a curved bacilli. Okay. 1 second. I have a question here. Ma'am, when do we have to wash? Okay, acha, washing. Washing is after every step. Okay, I'm sorry, I can go through the steps again. I think I forgot to mention that. Okay, I'll mention it. And then I'll help you remember कि कौन से ऐसे staining में हम wash भी नहीं करते. Let me go back to that. Okay, so let me just complete. Okay. So you'll be able to see rounded ends and no particular arrangement, because when you are seeing gram-positive organisms, especially gram-positive foci like Staphylococcus, you'll be able to see grape-like structure. And this uniformly stained, of course, is outside your scope of understanding right now. But there are certain organisms that are not uniformly stained on gram stain, and they have medical importance, causing very severe diseases. So it is very important to see whether the organisms are uniformly stained or not. Okay, just going back to that. Very quick revision in 2 minutes. Crystal violet add kiya humne, then you wash. Crystal violet is added for 2 minutes. You wash the slide under tap water. ठीक है. In the exams, if you are given gram staining to do, तो एक beaker या किसी container में ना add, put some water in it and keep it, and then use that to wash the slide, so that your smear is not run away. ठीक है, along with the water. Then you add iodine, grams iodine we are adding. That is kept for 2 minutes. Then you wash again. फिर आपने add किया acetone for 2 to 3 seconds. Usually 2 to 4 बोलते हैं. Never write in odd numbers. Then you wash the slide again. You add safranin, 0.25%. You are keeping it for 1 minute. Then you are washing the slide. Then you are blotting it dry. Blotting dry कैसे करते हैं? You keep the slide. You have blotting paper. ठीक है, ये blotting paper का एक folded part है. You keep the slide here. You fold this over the slide and tap it, and then your slide will be blotted dry. And then under oil immersion lens of the light microscope, this is 100x, you are going to see the stain slide. Now, if you all remember something known as Albert stain, done for Corynebacterium diphtheria, उसमें you do not wash. There is no washing in the steps. Albert 1 और Albert 2 के बीच में कोई washing नहीं होता. And even after Albert 2, you have to simply blot dry the slide and see it under the microscope. Then there's another one that you all do in the exam. Nielsen staining. इसमें you are first adding the carbon fusion. Then you are intermittently heating it. Then you are adding 20% H2SO4, that is sulfuric acid, till this color of the carbon fusion runs off. And then you are adding either malachite green or methylene blue as a counter stain. In between H2SO4 and methylene blue, you can wash, but after methylene blue and malachite green, you have to wash the right and then blot dry it and observe it under the light. Okay. Any questions? Any confusion? gram stain में बहुत questions पूछे जा सकते हैं. So you, even if it seems like a very small topic, please. Any questions? Okay, can I repeat the statement for bacteria? अच्छा, वो जो last में exam में लिखोगे. ठीक. Okay, so I'll just tell you gram positive अगर आता है आपके पास, so you all will write gram positive cocci. Supposing you get, okay, let me write a general statement. Gram positive cocci seen in clusters, या फिर pairs, या फिर chains, whichever structure you are seeing, uniformly stained. This is for gram positive cocci. ठीक है. And for gram negative bacilli, at your level, this much of the statement is enough. For gram negative bacilli seen with parallel sides, and round ends, uniformly stained. With no particular arrangement. Now you understand why the statement of arrangement is important, because gram positive में हम arrangement पे comment करते हैं, and that is how we decide the further biochemical tests that we have to do. Ma'am, uniform, non-uniform stainless differentiate between live from dead bacteria. But not in all cases. You can't really say. Okay, actually, if you're talking in respect to tuberculous wale bacteria, bacilli, when they are dead, they are not really not uniformly stained, but they become small and stout. They don't give that characteristic beaded appearance. But you can't differentiate between live and dead bacteria on a gram stain because when you're staining the organism, when you're heat fixing the slide, the organism automatically dies. So don't gram stain. Chalo, I think any more questions or should we end the class? Okay. So thank you so much for paying attention and sorry for the glitch in the beginning. I hope this is clear. Okay. Chalo, bye-bye.